but I think we should move on to the next talk. That one is by a postdoc in the group, Ariel Aptekmann. Ariel had received his PhD in biological chemistry from the University of Buenos Aires in 2018. And since then, Ariel is a postdoc with the ENIGMA project, currently working with the Ferrero and the Bromberg labs, where he's developing machine learning-based algorithms to elucidate protein properties and molecular functions. He's going to talk a little bit about his current project, I hope, to build a computational tool for predicting protein metal binding from sequence alone. Ariel?
Hi. Thanks for the introduction, Jana, and thanks for inviting me. Well, so the title of this talk is Mebipred, metal binding prediction for protein sequences. Mebipred stands for metal binding predictor. It is a tool based on machine learning that I have developed and refined during the last 2 years. And Mebipred can predict the binding of 10 different ions on protein sequences. Mebipred is very precise. It's very precise in identifying protein sequences that are likely to bind the ions listed on the y-axis. Let's take, for example, the zinc, the first row. The blue bar shows that at the default cutoff, Mebipred has a precision of 95%, predicting zinc binding from protein sequences, much higher than the precision expected by a random classifier, which is around 5%. And that's represented by the red bar. Mebipred is better than a random classifier for predicting zinc binding proteins from sequence, and the same thing applies to all the other metal ions that are presented on the y-axis. It is also worth noticing that Mebipred would also identify ligands containing one of these ions as binding the ion itself. For example, it identifies heme binding proteins as iron binding. Mebipred also happens to be one of the only available methods for predicting metal binding from protein sequences. While there are methods that predict metal binding from proteins from their structure, there is only one other method that predicts from sequence, at least that we could find. It is metal detector 2. Mebipred performs better than metal detector 2. Let's take, for example, the first row of this table. At the same recall on the same dataset, Mebipred has a precision of 90%, while metal detector 2 has a precision of 63%. Mebipred has performed better than metal detector 2 on all of the datasets they used for testing. As a final note on Mebipred's performance, Mebipred is really, really fast. It is definitely faster than methods that use structure or sequence alignment for their predictions. And when I say really fast, I mean several orders of magnitude faster. It can predict the whole SwissProt database in about 30 minutes. It can predict the whole PDB in around 5. So because Mebipred is fast, it can process large amounts of data quickly. This means that Mebipred can improve database annotations. Let's consider, for example, two of the largest and most influential protein databases, PDB and SwissProt. There are around 250,000 sequences that are shared between both databases, from which around 50,000 have differences in metal binding annotation. That's around 20% of the proteins on both databases. Of these discrepancies, around 61% are proteins that have a metal in PDB, but no metal annotation on SwissProt. The remaining 39% are proteins annotated as metal binding on SwissProt, but that don't have a metal on PDB, in PDB. So we ran Mebipred on these discrepancies, and we happily observed that Mebipred mostly agrees with the positive annotations for both databases. That's really good because usually positive evidence has more weight than negative one. So I further tested Mebipred on metagenomic samples and observed that predictions for metal binding inferred from metagenomes follow the environmental trends. We first ran Mebipred on the third metagenomic samples, marine metagenomic samples that were graciously provided by John Bongiorno and Donato Giovanelli. I don't know if they are here, but thank you for providing the samples. And we found that the predictions of iron binding inferred from these metagenomes reflect the environmental iron concentration. We observed a similar pattern for zinc and manganese, but not for other metals. We then tested Mebipred on metagenomic samples coming from different depths at the same site on the Black Sea. And this is very interesting because the Black Sea is strongly stratified, which means that the ion concentrations change with depth. And thank you, Mihaela Klamotzicha, for providing us help with knowing the concentrations at different depths, because that was not so easy to find out. And along with other things that changed, like pH and salinity, the concentrations of the ions that we predict also changed. Closer to the surface, iron concentrations are lower and calcium and magnesium concentrations are higher. As I expected, on this figure, we can see that on the surface, there are less iron binding proteins and more calcium and magnesium binding proteins. This trend reverses itself as we go deeper. We also found that the largest difference between two depths is the one between 50 meters and 170 meters. This can be observed on the black line that's labeled Euclidean distance, and it can be explained by the fact that around 100 meters is the photosynthetic limit, which means that the microbial community changes drastically. So far, we have talked about marine metagenomic samples, either on the Black Sea or the marine samples obtained from the deep sea sites that Joy provided. In line with these findings, we also observe that different environments have different metal binding profiles across host and soil. And that takes us to the next slide. So different environments have different metal binding profiles. On this figure, the different colors indicate different metals, and each column is a different metagenome. We can observe that the more diverse environments, gut and soil, are also the most colorful ones, which means that they have more diversity in metal binding proteins. It is also interesting that for the samples coming from humans, not the gut one, the other ones, there is almost no significant prediction of iron binding, with the exception of one vaginal sample, which can be explained due to menses. Well, Mebipred is currently available online at services.brombolive.org/Mebipred. And while it is still on a beta version, you are more than welcome to try it out, and we would really appreciate feedback. For the more adventurous, Mebipred is also available as a Python package called MyMetal, which can be installed via console using the pip package manager. I would like to thank Rutgers and NASA and NIA for the funding, all the members of the ENIGMA team, Joy, Donato, and Mihaela, who helped with their data and resources. And finally, thank Diego and Yana, who are my co-advisors for this project. And all of you for your attention. Thank you.
Thank you, Ariel. Are there any questions? If there aren't, I just want to point out that for a minute that you could, in fact, predict metagenome data that Ariel showed, but didn't actually explicitly mention. So you could submit your metagenomes and we will annotate short reads. Geoffrey Siwa asks, how does Mebipred perform for proteins that bind multiple metals with comparable affinity?
It predicts both metals as binding. For example, I remember one of the talks yesterday that talked about nitrogenases, and the first thing I did was go in there and started running predictions for nitrogenases. And it turns out that it predicts with the highest score, the one that, like, for example, the iron nitrogenases predict iron with the highest score, but it also predicts other ions as binding too. So it predicts multiple metals as binding.
Great. Any other questions? What about false positives? Asks Drawer.
What about false positives? Well, there are false positives, definitely. But what do you mean? Could you be a little bit more specific?
I guess if the question refers to false positive in the sense of multiple metal binding, how do you know that the second metal is not.
I'm actually I'm actually asking about sequences that shows up as metal binders that are not metal binders. How do you check that?
How do we check that? Well, those are around 10%. Like it has a really low percentage of false positives, but there are definitely some. And in some cases, when I checked on those false positives, it has something to do with it being a fragment of a larger protein that actually binds a metal. So it's picking up some signal from there.
Okay, thank you, Ariel.
Thank you very much, everybody. Berta, Jana, Ariel.